chapter 14 and 15, we will speak on how our microorganisms are involved in causing disease. Many of the topics here include a number of terminologies that we use to associate with microorganisms in causing disease. So that will be our focus, microorganisms causing disease. And many of the topics that we speak on are general to different microorganisms, whether bacteria, we're talking about viruses, protozoan, helminths, all right? They will infect cells and cause disease. So we'll look at some of them here. In chapter 14, we will look at many terminologies associated with disease. So principle of diseases, all right? So in looking at our diseases, one group of organisms we have to, again, familiarize ourselves with is the normal microbiota. And these are organisms that live with us during certain periods of our life. And the population of the normal microbiota will not always be the same throughout our lives. And those organisms that may have helped us when we were little kids might be slightly different today, right? So maybe it's slightly changed or is completely different. But these organisms of the normal microbiota, they either permanently, transiently means that they're there for periods of time. So maybe they were there during certain ages of our life in order to help us. And these organisms will be found in various regions of the body under normal conditions, right? And the normal microbiota, they will vary, right? Those organisms that are found in the skin will be different from those organisms that are found in the respiratory tract. Those organisms in the respiratory tract and skin will become completely different from those in the stomach, right? Different organisms occupy different areas of the body, and those will be their normal habitat, normal conditions, right? So certain conditions, right? The skin is a very harsh environment and is not a great place for many organisms to be. So you have, in order to survive the skin, you have to be equipped to do so. And in order to survive, let's say, the stomach or the small intestines, you have to be equipped to survive those areas. The normal microbiota, as we learned, they are typically harmless and they are there to protect us, right? So we will mention them again in class when we talk about immunity. They are there to protect us. One thing we need to know about the normal microbiota in respect to our cells is that the large intestines have the most microbiota, not just the largest number, but even the highest diversity in the normal microbiota population. And what do you propose these organisms are doing there? Can you remember why they are there in this area? Well, for one, they help us to absorb nutrients. Right? Two, they help to aid in digestion, right? So they help us to digest substances, right? These small organisms, they have metabolisms, enzymes that can break down things that we can't do without them. Some of them, as we know, help us to get vitamins. Right? So this is all, these are some of the things that these organisms in the normal microbiota will be doing. All right, so let's look at the large intestine. The large intestine has Escherichia. You also want to go over your enterics, right? There's so many enterics that we talked about that will be found in humans. What are they? All right, so you need to go back to chapter 11, all right, where we talked about these enterics, all right? So there's many enterics. You should go over the shape of these organisms, what they look like, what is the classification of these organisms in chapter 11. All right. So on the skin, we will find mostly Staphylococcus. All right. So those are the predominant organisms on the skin. Staphylococcus, mostly on the skin. They can be found in other regions of the body, but predominantly the skin if you want to isolate them. For the respiratory tract, the upper respiratory tract, we have Streptococcus. we can find those also predominant. Right, another organism on the skin that we talked about, Corynebacterium. Right, so go over these organisms, go back to chapter 11. We talked about whether they're gram-positive, gram-negative, right? Whether they're part of proteobacteria, non-proteobacteria, Firmicutes, actinobacteria. You need to know what they are, all right, going forward. So if you don't remember, this will be a good time to go back, put a little note on the side of your slide, so that way it can be helpful as you study. So these pictures show us different organisms that are going to be found in the body. So let's look at picture A. Picture A shows us bacteria on the surface of the nasal epithelium. All right, so these are round-shaped organisms on the nasal epithelium. All right, let's say that I said that they form chains. They are gram-positive, and they're in phylum Firmicutes. They are aerotolerant. What is this organism? Right? Will you be able to figure it out? And for B, so see if you can figure it out. You have to go back to chapter 11. For B, these are bacteria on the lining of the stomach. So let's say that I say that these are curved rods. They are gram-negative. They have peritrichous flagella. And these organisms are in class, so the group that they are in would be class Epsilon Proteobacteria. What are these organisms in the lining of the stomach in B? And now in the small intestine, we have these organisms here. All right, so let's say perhaps I said that these are going to be gram-negative, right? Let's say that they are rods. We'll say that these are enterics, just to use it as an example. Peritrichous flagella. Facultative. Anaerobes. In humans, what can these potentially be? All right, so you should know the characteristics of these organisms as you move forward. If you're not sure of what these can be, aerotolerant. You should go back to chapter 11 so that way you can figure them out. All right, so just get an idea of all these characteristics. All right, and some of these we will review in class as we move forward. Okay, but we're talking about our normal microbiota. It's very important to talk about these organisms when we're talking about disease, right? Why? Why do we need to talk about these organisms? So as we move forward, we will get to see why is it important to talk about these organisms and causing disease? Because normally they are there to help us, all right? So why do we need to talk about them causing the diseases? So the normal microbiota, they are found in different areas of the body. As I mentioned, different areas of the body will have different populations and varieties of these microorganisms to protect you. But what are they doing? And these are four mechanisms that discuss how these organisms will help you, all right? So number one, the normal microbiota will create a barrier, right? So they're like an army and they will protect you. So this is your army, right? They create a barrier or defense protecting you. You are their territory that they're trying to protect. Number two, they will compete with the pathogen for nutrients. So by exhausting all nutrients, the pathogen can't have much to survive. And if the pathogen doesn't have much to survive, right, then they'll die, right? So this is what they are doing. Number three, some of them will change the environment where the pathogen will occupy. And one way that they can do this is by changing the environment to a more acidic environment. Most pathogens are neutrophils and they do not survive acid. So we have some organisms on the skin, such as Staphylococcus, so I'll just put staph. We'll put Propionibacterium. And we have Corynebacterium. And these organisms occupy the skin. And one thing they do is that they increase the acidity of the skin, all right? And how do they increase the acidity, this environment on the skin? What they do is they make an enzyme called lipase. Okay, what does that enzyme do? What does it digest? So it digests fats. So any fat on the skin, and we call that fat, right, sebum, on the skin, the oil, right, it will digest it. And when it digests the fat on the skin, it will make two components, right? What are the two monomers of the fat, of a fat? Well, glycerol is one, and the other one is your fatty acid. Right? So this is, as they break down the sebum, so they break down the sebum on the skin using lipase, and the fatty acids will grow up. Right? So the environment of the skin is very acidic. A lot of organisms can't survive that. They can't even get past the skin to get to other areas of the body. So that's one way that these organisms, 3 gram-positive organisms, create an environment to help you. Some bacterial cells produce toxins. We call these bacterial sins, and these toxins will lyse the pathogen, right? So they can be harmful to the pathogen. So these are different ways that the normal microbiota are there to protect you. So they're not just sitting there where they're not doing anything. They actually use one of these strategies, one of these 4 strategies, in order to protect you. So again, why are we talking about these normal microbiota, especially in diseases? Well, the normal microbiota, they can become what we call opportunistic pathogen, right? And what does this mean? Exactly what it says. Well, these organisms will infect you when they have a grand opportunity to do so, all right? So when is that opportunity? That opportunity is usually when your immune system is not so well, and they can infect you. So the normal microbiota, they can take advantage of a compromised, a weakened immune system, an impaired immune system, right? So this immune system is weak. So what do we mean by weak? This means that your immune system is not doing or is not functioning at its maximum capacity at that time. And this presents an opportunity for these organisms that will normally help you. Now they become harmful to you, right? And they start to invade the skin, the respiratory tract, and other areas of your body. So what makes a person compromised? There's a number of factors that can make a person compromised or impaired. What may change or weaken the immune system? What are some of those factors? Right? Well, there's a large number of factors that can change your immune system. So some of them, I'll name some of them. One is stress, right? We'll put lack of sleep, right? If you're exhaustion, right? Let's say poor nutrients, right? So if you're not eating well, all right? If you're sick, right? If you're sick with some other disease or some other illness, these organisms may invade you. So if you have a disease or infection, So if you have another disease or infection, these organisms can infect you. So any changes to your immune system, which can be due to environmental factors, can be due to many different things. It will make if your immune system becomes impaired or changed in some way, it allows these organisms that are naturally there to protect you now a space and an opportunity to invade. So besides the normal microbiota relationship, there's a number of relationships that our microorganisms can have with their host. So where does the normal microbiota fit? The normal microbiota will fit in more or less these two categories, either of mutualism and commensalism. So we'll put normal microbiota, where these organisms, they're living with the host where both the host and the organism benefit, or the commensal relationship is when one party benefits and the other is not harmed. So that's usually where the normal microbiota fits. And in your book, they give you two examples. For commensalism, they show you the relationship of Staphylococcus being in the host. And for mutualism, they show you the relationship of E. coli in the gut. Right? So this is where the E. coli would be. Right? So parasitism, usually when we're talking about parasitism, one party is harmed to the other party's benefit, usually will involve something that is a pathogen. All right, sometimes even a parasite. So they give you H1N1, right? So what are they talking about? H1N1, that will be your flu, right? So one of your flu viruses, and they are going to be harming the host. All right, so those are your symbiotic relationships. Symbiotic means that two parties are living together for a period of time, right? Or it could be a long period of time, like when we're talking about endosymbiosis, right, with the mitochondria and the host, the eukaryotic cell, or it can be just for a short period of time. Okay, so what are some other relationships that microbes can have with their host? Probiotic, right? So what is a probiotic? Right? If you're into health, you may know, or if you exercise, you go to the gym, you may have heard about probiotics, or maybe even from a commercial. Probiotics, these are microbial cultures, and what you're going to do is ingest them. You take them in, and the goal is when you ingest them, they will have a benefit where? Where would most of their benefit be? In the digestive tract, because that's where they're coming in, right? So mostly by the time these organisms get through the stomach and get to the digestive tract, get to the intestines, only a small amount of them will make it to the intestines where it can benefit your gut health, right? So again, you have to ingest these probiotics. They have to be ingested, and you're ingesting them for a benefit, right? You're trying to get benefit for some of them. So if you, what are some examples of probiotics? Right, so these include bacteria like lactobacillus. Right, so lactobacillus found in yogurt. You want to, again, go back to chapter 11 to review the characteristics of lactobacillus, what it looks like, what phylum it's part of. And when you ingest these lactobacillus, usually there's, in these probiotics, there's millions of these bacterial cells that you're ingesting. And only some of them will get to the large intestine where they will contribute to creating a healthy environment, right? So maybe your gut from eating foods for so long, it's not so healthy, it's toxic. So the goal is that we want to get these good organisms, the ones that are in the yogurt, right? They haven't been exposed to the environment of your stomach yet, or environment of your gut yet, and we want to ingest them, and hopefully they will help to create, a healthier environment for the cells to grow in your gut and digest food. So our other microbial relationship we have is microbial antagonism, and this is competition. So that means that the organisms that are present, they're competing against each other, and this competition is going to create some sort of equilibrium. balance. All right, so it creates an equilibrium. So one organism is going to check the other one, right? And keep it so that way they're not growing so much. So one of the best examples of microbial antagonism in our body is the relationship between the bacteria, such as lactobacillus, and yeast, like candida, right? So we talked about candida, which can cause yeast infection. And by having these organisms in competition, they keep each other in check. When one of them go out of balance, the other one then starts to grow a lot, and it will cause you to get sick. So in the case of our bacteria and yeast that live with us, if they're out of balance, there's no disease. But if the yeast population goes down and the bacteria go up, you can get some sort of bacterial infection, right? But again, if they are balanced, right? So if they are balanced, then nothing will happen. And the opposite can happen if the bacteria population declines, and the yeast will then be will be unchecked. So the yeast then will start to grow and grow and grow, and that will cause some sort of yeast infection. Right? So that gives you an idea of microbial antagonism. Again, organisms have to be in balance, and they will check one another, keep them growing at a certain level. Right? So one doesn't overpopulate and make the whole sick. In chapter 14, they talk about Koch, all right? So Robert Koch we have, and Koch is important because he showed us something very important. Koch proved microorganism can cause a disease, all right? So he showed that microorganisms can cause a disease, and he did this with two organisms. He used Mycobacterium tuberculosis. Right, so any organism he infected with Mycobacterium tuberculosis, well, they started to cough, lung infection. Right, they'll get very sick, fever, lung infection, cough. And the other organism he worked with was Bacillus anthracis. So we'll put this as Koch's test microbes. Right, so one was Mycobacterium tuberculosis, and the other one Bacillus anthracis. And every time that these organisms were infected, well, the cardiovascular system was damaged, and many of them will die. All right, so these individuals that were infected with Bacillus anthracis will get anthrax. So those are the organisms he worked with, right? So how did Koch show that a microorganism can cause a disease, all right? So what did he do? All right, so these are the steps. It shows you what he did, and this is very important here, okay? So in step 1, it says the microorganisms he isolated from an animal that was already sick. So they were already sick with either Mycobacterium tuberculosis or Bacillus anthracis. So what he did is that he took the microbes from the rat that was already dead, right? So he took those microorganisms. How did he take them? Well, he'll take a blood sample from this microorganism, let's say, right? So some sort of blood sample that was already contaminated with the microorganisms because this animal had died. So what did he do? In the next step, which is 2A, the microorganisms, he grew them in pure culture. So he viewed and grew the microbe, right? So why was this important for him to grow the organisms in pure culture? Why? Why did he grow the organism in pure culture? Right? What does it mean, pure culture? Well, pure culture means that you want to make sure that there's only one microorganism. Right, so he would grow it in the laboratory on some sort of nutrient agar, and he would verify that in that blood sample that there wasn't 3 organisms causing the disease, right? It was only 1 organism. So he would grow the microorganisms on a petri dish until he would have only 1 specific colony, all right? So if he saw, let's say on the petri dish, 1 colony was yellow and the other colony was white, he would know that on that petri dish, well, there's a mixture of microbes. So what he would do, he would take one of the yellow colonies, leave the white one on the dish, and then grow it on another petri dish to make sure that he would only have the yellow colony, right? So that way he's very specific for the organism that he will be talking about. So the other thing that he did is that he identified these organisms under the microscope. So if it's bacillus or mycobacterium, he would see rods, right? So he knows what this organism will look like, this microorganism. So what did he do? In step 3, he took those microorganisms and inoculated. Inoculate means that he injected, right, or he grew it in a nutrient environment. So he grew it in a healthy animal. So he injected it, we'll call this into rat number 2. All right, so this is rat number 1. This is the one that is already dead. This is rat number 2, right? Right now he's healthy, but when he injected the microorganisms, and not the microorganisms from anywhere, the microorganisms from the pure culture, right? If he took the microorganisms from the blood, we don't know how many other microorganisms are in the blood, but he took it from the pure culture and injected it into this rat. So what happened? The poor rat died, right? So now, does he know at this point whether or not which microorganism made this rat sick? No. So what did he do to confirm it? In step 5A, the microorganisms are isolated from this animal. From which animal? From number 2, right? That has now died, and he will grow it in pure culture. So what he wants to see is that whatever organism grows here is identical to the yellow one, or whatever organism he saw in step 2A. And he also made sure that the organism that he saw in rat 2 should look the same, right? So he verified that these are the same organisms that the one that he saw in the case of rat 1 that killed rat 1, right? So rat 2 showed the same signs and symptoms as rat 1. It had the same microorganism, so he knows that that microorganism caused that disease. The other thing that Koch showed is that microorganisms can be transmitted, all right? So he showed that he can take microorganisms from one animal, right, and then put it sort of into the next animal, and that will get them sick, right? So the most important thing here is that Koch showed that microorganisms cause disease, but what's also important, or a secondary finding, is that he showed that microorganisms can be transmitted, right? So these are some things that Koch did. Okay, so there are some exceptions to Koch's postulates. This doesn't mean that what Koch stated isn't true. It's absolutely true, but it doesn't always apply, all right? So these are some of them. So number 1, right? So this is the first one. Some organisms cannot be grown in media, right? So some organisms we cannot grow in nutrient agar. We have to grow them in a special environment. So it's not as simple as bringing them into the lab and they're growing on pure culture, right? So that's one exception. And Mycobacterium leprae is an example of that, right? So Mycobacterium usually needs to be grown with whole cells. So you have to grow with whole cells. All right, so special requirements for growth. Number 2, one of his exceptions is that one disease can be caused by multiple microorganisms. So in the case of, let's say, Bacillus anthracis, anytime you infect a host with this bacteria, you would see the same sign and symptom. But in the case of pneumonia, it could be many different organisms that can cause pneumonia. I mentioned 2 bacteria here, but there can be viruses that can cause pneumonia, fungi, protozoan. There's so many different organisms that can show the symptoms of pneumonia. All right. So number 3, one microorganism can cause multiple diseases. All right. So Streptococcus pneumoniae, for example, if it infects a host, it can cause pneumonia if it infects the lungs. Meningitis if it infects the brain. Ear infection. So depending on where it travels, it can cause different infections. So you may not always see the same sign and symptom when Streptococcus or some organisms are infected into their host. Number four, right? Pathogens can cause disease in only certain hosts. All right, so something like HIV, if you think you're going to use a needle and transmit it to a rat, you're not going to see the same signs and symptoms of a disease that you would see in humans, right? Because humans is the host that the HIV requires in order to show how it causes the disease. And the last one, I added this in, number 5. This is not a true exception, but not all diseases are communicable. Okay? So in being communicable, this means that not all diseases are transmitted. They're not all transferred from one subject to the next. So this is not a true exception, but I did add it in here. All right, so communicable, again, means that they can be transferred from one host to the next. All right, some diseases are non-communicable. So stages of a disease, right? Each disease goes through different stages, and the stages that the disease goes through, we have this graph here where the number of microbes increases, so it goes up, all right? So it's going up here, all right? And this is to reflect how the disease progresses. So the time is the progression of the disease. So it starts out as the incubation period, where we have a low number of microbes, and then the next period will be the prodromal period, the period of illness, where we have the highest number of microbes, so the number of microbes will increase, right? So it's going up, right, up, and then we go to a period of decline and the convalescent period, right? So does the trend of the stages of disease look like a curve that we covered earlier, a graph that we covered earlier? What graph would that be? Well, we talked earlier about the phases of bacterial growth. Or microbial growth. All right, where each bacteria, right, they go through a number of these phases. All right, so over time, the number of microbes will increase. So we'll put number. All right, so it will look something maybe like that, right? And the terms that we use to describe these phases, we had log, lag, death or decline, and then we had stationary. We'll label it as A, B, C, or D. All right, so how would we label our curve here for the phases of microbial growth? Anybody remember? All right, so where do we put our lag phase? All right, so lag will be the first part, right? Where there's not just like in this stage of disease, not that many microbes. And then where would the log go? Well, that's when we have most of the cells growing, right? And that also corresponds with our period of illness, all right? And then we have our stationary, and then of course over here would be our decline, all right? So the stages of disease match up with the phases of microbial growth. Okay, so let's look at the different periods.